on genetic engineering. So our aim today is to explain the techniques and principles used in genetic engineering. And before we even begin, I've got to say there's a lot of different techniques and principles used in genetic engineering. So I think the main priority is to be aware of key enzymes and key definitions, and to have an outline of the different techniques. You don't need to know full detail about every single one, but I'll highlight the one or two ones that I think are most likely to come up in an exam question. So before we really get started, I'd like you to just pause this video and make a note of these key definitions here, and then we'll get started with our sort of overall principles of genetic engineering. Okay, so I think it's worth talking about why genetic engineering. Show you some interesting pictures first. So here's some pictures of some genetically engineered organisms that have been engineered with a gene called green fluorescent protein. Now, this is more a proof of principle. So this is a marker gene. This fluorescent gene can be introduced to see if we are successfully engineering an organism. We've got a rat here. We've got some vesicles. I think those are the lysosomes highlighted there with the green fluorescent protein in some cells. And then here we've got, this looks like the vascular tissue, possibly the xylem of a plant. We can also genetically engineer crops to have better nutritional content. This is golden rice, which has been engineered to have a higher vitamin A content, reducing the likelihood of vitamin deficiencies. And we're going to look at more of these examples in the next lesson. But I thought it'd be interesting to show why do this before we say how. Here's another example, again, more in this next lesson. We're going to be looking at the applications of this technology, and now let's get into the how. So, the overall principles of genetic engineering are, first of all, to identify the gene that you want to put into a new organism. Once you've identified that gene, you can produce multiple copies of it. You can then try and put it into a vector. Now, a vector means something that can carry a gene from one place to another, and then you have to transfer that gene into the cells that you want to engineer. Once you've put the gene in the cells, the next step is you have to actually be sure that they've taken up the gene, and you do that using a marker, which could be to do with antibiotic resistance, or it could be some other techniques. And then finally, we allow the host cells to multiply, or we clone them up, or we grow them to produce whatever we want to. So we're going to look at these four key phases of genetic engineering one at a time. So we're going to start with how do we get multiple copies of the desired gene. So here are the four different techniques that you need to be aware of. You'll find some more information on page 228 of your textbook about these, but I'm just going to talk you through each one. So first of all, messenger RNA from a cell can be turned into cDNA. What does that mean? So messenger RNA, remember, is made when a gene is transcribed, and the messenger RNA exits the nucleus. It's already been edited, so it's had any introns removed, and it exits the nucleus. Well, what we can do is we can use an enzyme called reverse transcriptase. And what happens there is reverse transcriptase binds to the messenger RNA and it turns it back into DNA. Okay, here we've got a hybrid, and then along comes another step. So it takes that kind of hybrid mRNA and turns it into double-stranded cDNA. Here's the process here. So once we remove this first bit of messenger RNA, we can turn it into double-stranded DNA, which looks like this. Okay, so we take single-stranded messenger RNA and we convert it into double-stranded DNA, and that uses an enzyme called reverse transcriptase that we actually isolate from retroviruses like HIV. Okay, so that's one technique. Well, if we already know the sequence of the gene that we want, we can also use primers to replicate it by PCR. Okay, so we looked at PCR 2 lessons ago. Here's a little recap. So the primers are those orange bits there, and using primers we can really focus in on the part of the DNA sequence that we want to copy, and we can amplify it many, many millions of times to produce lots of the gene that we want. So that's PCR. Another technique we can use is we can use a DNA printer if the code of the gene is known. Pretty much if it's in a computer typed out as letters, a sequence of letters, we actually have machines that can print that, and those machines have come a long way in the last sort of 10 years, and it prints the gene which we can then use. So you don't need to know much at all about how this works, but here's a little image showing that we have the sequence, the printer prints it, can print it on a chip, for example, and then we can use that and put it into a vector. Finally, we can use restriction enzymes to cut out a gene from a longer stretch of DNA. And restriction enzymes cut at specific sites. So we're going to look at some of these methods now in a bit more detail. Okay, first of all, we're going to look at producing DNA copies from messenger RNA with that reverse transcriptase enzyme. So here we are. Here we have a cell. We see the nucleus here, the rough endoplasmic reticulum, etc. So messenger RNA can be isolated by taking the cell, breaking the cell open, and then centrifuging, spinning the cell at a certain revolution. This is 10,000 G. And then we can find in a certain layer the messenger RNA. Then we can take the enzyme reverse transcriptase, here it is, and that will basically turn the messenger RNA back into complementary DNA, which is called cDNA. And then we have just a little further step, which kind of removes the original messenger RNA. You don't need to know the details of that. Leaving single-stranded DNA, and then we can convert that back into double-stranded DNA without the intron. So that's one key thing, is that the intron segments will be removed because we're using the messenger RNA to produce the new DNA. So we've already kind of edited it. The ribozyme, if you remember that word, has already edited the messenger RNA. So that's the first technique, using reverse transcriptase. What about the next technique? The next technique is using restriction endonucleases. So we'll first take you through this animation, and then we're going to label a sheet which you should have on the PowerPoint attached in Teams. You'll need to print that sheet, the first 2 slides of the PowerPoint that's going to be attached. Okay, so here is a restriction enzyme about to cut this piece of DNA. So a restriction enzyme cuts when it recognizes a specific sequence in the DNA strand. And it normally recognizes a site that we would call palindromic. So a palindrome is a word like, or 2 words like race car, that is the same spelt forwards and backwards. So race car, if you write it out, you can write it forwards, and then backwards it would say the same thing. So in DNA, when we talk about palindromes, we say that on one strand, the sequence going, let's say, this way is the same as the sequence going the other way on the opposite strand. So this enzyme is going to be looking for GAA-TTC. So we can scan along the DNA and find that sequence here. Then furthermore, it says that this enzyme cuts between the guanine and the adenine. So it's going to cut there, and it's going to cut there. So when it cuts like that, it's going to produce these overlapping sticky ends. So a sort of short segment of a single-stranded DNA there. It's going to cut there as well. So this would be a gene now which has been cut out by a restriction enzyme that has sticky ends. So different restriction enzymes work at different palindromic recognition sites. Let's have a look at one more. So this one is going to cut with this different recognition site, and it won't. If we cut the first gene with the EcoR1 and the second one with the Hind1, they won't align. So if we want to get a gene to go into a plasmid, for example, we have to cut the plasmid with the same restriction enzyme that we cut the gene we want to insert, because then those sticky ends will be complementary and then they will join up. Okay, so let's go back to our main slides. So now I'm going to take you through this sheet, which hopefully you've already printed out, and we're going to look at that in a bit more detail. So I want you to be labeling this as we go along. So to start at the top, we've got the enlargement to show the restriction site for restriction enzyme EcoR1. We saw it cut between the G and the A and the G and the A. So the DNA phosphate backbone has cut the positions marked by these arrows. And that produces this overlapping kind of single-stranded bit, which we call a sticky end with unpaired bases. And if we join DNA from two different sources, if they've both been cut with the same restriction enzyme, then those kind of overlapping, I'm trying to use my fingers, those overlapping ends kind of align like that, and they will join. So that involves complementary base pairing between the sticky ends. But when we've got that sort of join there, like that, there's still a gap in the backbone that we need to fix. So we're using an enzyme called ligase, which joins the sugar phosphate backbone. So if you look closely, it would be here. This bond, this phosphodiester bond, would be made by a ligase, and also down here, this bond would be made by a ligase enzyme. So that is the process of using restriction enzymes to cut out a gene and then to put it into a new place. Okay, so here's a short little quiz for you to do, taken off the BoardWorks. Which process do these statements kind of refer to? So first of all, results in cDNA. What do you think? That's this one. Uses DNA polymerase. Well, that's actually going to be this one too. RNA as a template. That one. Contains introns. That's something going to be this one. Sticky ends. Cuts DNA from a genome. Let's see if I got this right. Yeah, okay. So now let's move on. We've just been talking so far about how do we get the genes, and we've got a few more bits to do. The next thing is inserting the gene into a vector and transferring it into host cells. So a vector is a place or a thing which can transfer a gene from one place to another. Anything that can carry or insert DNA into a host organism. So some examples might include a plasmid obtained from a bacteria. We're going to look at that in a bit more detail. Or it might include a viral vector. So first of all, let's talk about the viral vector, and then we're going to go into some bit more detail with the plasmid. So viral vector might be this virus, for example. Now, viruses, as hopefully you're aware, have sometimes DNA or sometimes RNA in the middle, but what we can do is we can edit out the DNA from the center of the virus and replace it with the DNA that we want the virus to put into a cell. So, and if it's a retrovirus vector, then what that retrovirus vector actually does is it's evolved and adapted to put that DNA from the viral capsid inside the virus to put it into the host genome and to integrate it using an enzyme called integrase into the genome. So it's kind of, we're using a tool which has been kind of produced for us through viral evolution. Okay, so plasmids, let's go into a bit more detail now. So plasmids are these circular parts of DNA that are found in bacteria. So when we use a plasmid as a vector, we have to cut the desired gene with a restriction enzyme, and that restriction enzyme has to be used, the same one, to cut the plasmid. And then we can put the desired gene back into the plasmid. We use ligase to join up these two different backbone bits that need to be joined. And then we can take this plasmid and insert it into a cell. Now we're going to look at this in a little bit more detail, how we can actually use antibiotic resistance to figure out which bacteria have taken up the plasmids. But when we take the plasmids and we want to put them back in the bacteria, we normally have to do something called heat shock, which we're going to look at later. So we're going to look at this a little bit later, so I'm just going to skip through this. So we've looked at using viral vectors and we've looked at using plasmids as vectors. What about transferring into the host cell? So this can be done in a few different ways. Oops, that shouldn't be there, never mind. So first of all, we use heat shock. So basically this means that if we have plasmids floating around outside of bacteria, and we basically have the bacteria in some very cold sort of ice bucket, we leave it there for about 30 minutes, then we very, very quickly, we shove that bacteria into a very hot place, 42 degrees C. The bacteria basically panics, it thinks it's going to die, and what it does then is it basically just takes up any DNA that's floating around. It's kind of a survival response. Basically, a bacteria are about to die anyway, they'll just grab genes from anywhere just to sort of see if it's going to help. So we can use this response, it's called heat shock, to kind of get the plasmids into the bacteria. So that's heat shock. Another method is called electroporation or electrofusion, and basically it applies an electric field to the bacteria. So what's shown down here is before a pulse of electricity, the DNA, which is these little green wiggly bits, is kind of outside the cell, and if we apply an electric field, and it causes these tiny holes to appear in the membrane temporarily, and what we hope is that some of these DNA molecules will get transported inside the cell during that pulse. And then afterwards, remember the membrane is a fluid mosaic, so it kind of has an ability to heal itself spontaneously after you turn off the electricity, and then hopefully you've got some of the DNA inside the cell. Now it's not that efficient, doesn't work very frequently, but if you're talking about millions and millions of cells and millions of molecules of DNA, a few of them will get in. Next one, transfection. This basically just means using something called a bacteriophage. So a bacteriophage is a virus that infects a bacteria. So you can see here that this is a bacteriophage. They look almost like little space invaders, and they have the viral DNA or RNA up in the capsid there. And what they do is they land on the bacteria and they actually inject the DNA down into the bacterial cell. So if we replace the DNA that they're injecting, or the RNA that they're injecting, then we can engineer bacteria using transfection, using bacteriophages. The final one is kind of a weird one called TI plasmid. Now this TI plasmid is kind of complicated, so we'll zoom in down here a little bit. It uses a bacteria to infect a plant cell. Now this bacteria is called Agrobacterium tumefaciens, and it is responsible for causing these kind of growths on trees that you may have noticed. This is called a crown gall. And the bacteria makes these growths basically because it kind of tricks the plants to grow food and produce this kind of house for the bacteria. So in that gall, it's just a big colony of bacteria that is getting food from the plant. So the way it does this is by engineering the cells of the plant in that area to grow into this kind of bacteria house, if you like, and to produce chemicals for the bacteria to eat. So it's already evolved this very sophisticated way of engineering plant cells, so we can kind of hijack that by changing the genes in the plasmid, in this TI plasmid, which is called the tumor-inducing plasmid. And if we change the genes in that plasmid, we can get our genes, or the ones that we want, into the plant's cells, as opposed to the genes that this bacteria wants to put in the plant. Okay, so those are 4 different methods of putting the gene into the host cells. Okay, so far we've covered identifying the genes, putting them in a vector, and transferring them into the host cells. But there's another thing which is quite tricky. So let's go. Now we have to make sure that the cells that have taken up the gene are the ones that we grow. We do this using a marker. So we're going to go through this sheet. Again, you'll need to have printed this out. If possible, because it's quite complicated. And this is a process called replica plating, and using antibiotic resistance genes to identify which cells have taken up the gene that you want. So here we have a plasmid isolated from a bacteria. Now this plasmid contains 2 resistance genes, ampicillin resistance and tetracycline resistance. So when this plasmid is in a bacteria working properly, a bacteria won't be destroyed by ampicillin and it won't be destroyed by tetracycline. Now when we engineer this plasmid and we introduce a human gene, for example the insulin gene, we cut using restriction enzymes in this part of the plasmid and we insert the human insulin gene. Okay, so that means that bacteria that take up this plasmid, which has not got the insulin gene in, well, they will have both functioning genes. They will be able to stand ampicillin and they'll be able to stand tetracycline. Bacteria that take up this plasmid, that's actually the one we want them to take up. they will only be able to grow in ampicillin, but if we put them in tetracycline, this yellow gene now has kind of been broken in two, so it won't work. So we put these plasmids with loads of bacteria, we heat shock them, and the bacteria take up one of these two plasmids, or maybe both even, but we don't know which bacteria have taken them up and which ones have taken up what. So then we transfer the bacteria that we heat shocked onto a plate of agar. Now this plate of agar contains the antibiotic ampicillin. So that means that any bacteria that have taken up either this plasmid or this plasmid will be able to grow. So they will take up either the non-recombinant plasmid or the recombinant plasmid, and they'll both be able to grow the ampicillin. So here, after pouring those heat-shocked bacteria onto this plate, we can see that we've got 6 colonies growing. Now comes the complicated bit. How do we identify which colonies have taken up the recombinant plasmid that we want and not the non-recombinant plasmid that we don't want? So we do a process called replica plates, and it basically involves using a sterile block of velvet. So here we have the plate, which is the one shown down here, okay? Then what we do is we take that plate and we tap it on a piece of black sterile velvet. So we transfer the bacteria to a felt block, sterile felt. Here we go. Here's the sterile felt, putting it on there, tapping it down to make sure that the bacteria in those colonies are now attached to the felt. Then we take a brand new plate, and that's shown here. We take a brand new plate which has got tetracycline agar, so that's this down here. So you can see here in this little GIF here, we take the new plate. This is what's happening right now. This is the bacteria on the agar. New plate goes on top, and then we tap it down. So now we've transferred the bacterial colonies that are on that sterile block of felt. We've transferred it onto the tetracycline agar. Now, what do we notice? We notice that not all 6 colonies that were on the ampicillin agar will grow on the tetracycline agar. In fact, it's only 3. It's only colonies, looks like 1, 2, and 5 grow on the tetracycline agar. Now, let's think about it. Which ones do we actually want? Well, we actually want the ones that don't grow on tetracycline. So we can kind of forget about 1, 2, and 5, these ones. We don't want those because they grew on tetracycline. And we actually want the colonies 3, 4, and 6. So now we go in and we take a little loop and we take out, let's say, colony 6 there. We pick it up and we drop it into a tank for that bacteria to grow. We know that that bacteria has taken up a recombinant plasmid. So that is called using antibiotic resistance to select the recombinant bacteria. It's worth saying at this point that using antibiotic resistance markers is not without risk, because bacteria naturally can pass plasmids between bacteria. This is called conjugation. Here's an image here. This thing here is called a pilus. And it's a little kind of hair-like structure that can pass a plasmid from one bacteria to another. And they do this quite a lot. And this is one reason that antibiotic resistance can spread around between different bacterial populations. Other genetic markers we can use if we don't want to use antibiotic resistance, we can use fluorescent markers like GFP and other enzyme markers that, for example, lactase we can use. The enzyme lactase. And when there's a special kind of type of agar where if the enzyme lactase is present, it will turn the agar blue. All right, let's do this final 2 kind of summary tasks here, looking at the enzymes that we've talked about. Can you match up these enzymes on the left with their with their descriptions of what they do on the right? Pause the video, see if you can match them up, and I'll give you the answers in a few seconds. Okay. Here you go. I won't go through those for you now. I'm sure you can read those if you need to. And let's move on into our final summary questions. Oh, sorry, one more activity. Here's another quick true or false for you to do. You can just write 1 to 5 in your book and see if you can get this true or false. Pause the video here. See how much you took in of all that. There's a lot of information in this lesson. Okay, here we go. So the first one, do restriction endonucleases cut at palindromic sequences? Yes, they do. Second one, plasmids are circular pieces of DNA and they only code for antibiotic resistance. Well, that's not true. They do code for other things as well. We often use plasmids which have antibiotic resistance genes on them, but they do also code for many other things. So what's a vector? Is it an organism that expresses the protein coded for by the inserted DNA? No. So a vector is a thing that carries a gene from one place to another. The description here, an organism that expresses the protein coded for by the inserted DNA, that is kind of a definition of what you would call a transgenic organism or a genetically modified organism. Okay, 4. When we're talking about the antibiotic resistance markers, do we want the bacteria that are resistant to 2 antibiotics? No, we don't. We want the bacteria that are only resistant to the 1 antibiotic, and the second gene has been disrupted by the insertion of that gene into the plasmid there. 5. This one is true. So reverse transcriptase produces the cDNA. Right, final questions now. Thanks for sticking with this lesson. There's been a lot of information. So answer these questions. Mark scheme coming up in a few seconds. Just pause the video and you're going to send me your work green penned after this. Here we go. So you know the drill. Mark your work here, making sure you're focusing on key terminology. And before we finish, we'll just go to the syllabus check to see, what does the syllabus say about how much detail do we really need to know about all of this? Well, I'm afraid it doesn't tell you that much. It just says this. You need to understand the principles of genetic engineering, to include the isolation of genes, the placing of these genes into another organism using vectors. And then you need to know specifically about restriction enzymes and plasmids and ligase. So I think those are the most important ones. Everything to do with restriction enzymes, plasmids, and ligase. But you do also need to know about electroporation, which basically just means zapping with electricity to make a cell full of holes. Okay, so that was the lesson on genetic engineering and the techniques involved to do so. Next lesson, we'll be looking at the kind of ethical considerations and more of the applications of this technology. Okay, so I hope you find that useful. You may need to watch that back again because there's loads and loads of stuff in there. And thanks very much. Bye-bye.